A very warm welcome to the Rector, Father Dr. Swibert de Silva, our Principal, Father Dr. Victor Lobo, the Registrar, Dr. Melvin Colasso, the Vice Principal Postgraduate Section, Dr. Rabai, my colleagues, Associate Professors, Dr. Vanita and Dr. Wajid, Assistant Professor and Vice Principal, Undergraduate Section, Dr. Jyoti, Assistant Professors, Dr. Poppy, Miss Ria and Miss Shalmali, and my dear students. In the last 22 years of the postgraduate department, we had the traditional method of inauguration, but this is the first virtual inauguration. This marks the start of a fresh academic year and events for the association. We wish the staff coordinator and assistant professor, Dr. Poppy Datta, and the office bearers good luck and a very fruitful year ahead. We are privileged to have among us Dr. Subhojit Sen, Ramalingaswamy Fellow, faculty at UMDAE Center for Excellence in Basic Science, School of Biological Science, Mumbai. I would like to take this opportunity to express my heartfelt gratitude to Dr. Subhojit for accepting our invitation and gracing this occasion. We are all eagerly waiting to hear you, sir. Your presence and talk will definitely ignite many young minds present. I wish the association a very successful year with a variety of events and hope that the platform provided will be useful to all. Once again, a hearty welcome to all. Have an enlightening and pleasant evening. Thank you.
Thank you, ma'am, for the warm welcome. I now request our registrar, Dr. Melvin Colasso, to say a few words.
Guest of honor and speaker for the evening, Dr. Subhojit Sen, Dr. Rabi, I'm not sure whether he is there as part of this audience, Dr. Poppy Datta, coordinator of PG studies in microbiology, Dr. Beatrice, the head of the Department of Microbiology, office bearers and members of the Microbiological Society, students and guests attending this inaugural program. Good evening to you all. All of us have got so used to virtual meetings and functions these days. I sometimes wonder whether we are willing to go back to our classrooms. It is true that we are living in difficult times, but this has challenged us to do things in a different way. A virtual inaugural program such as this has opened doors not only for staff and students, but also for your friends and relatives. Unlike last year, no one needs to make an effort to go to a hall to attend the inaugural function or the lecture. This pandemic has opened up lots of opportunities, and I'm sure you must have made best use of the free time that was available. Just last month, I read an article about a 10-year-old boy studying in a school in Uttar Pradesh. The school, I think, is Presidium School in Uttar Pradesh. This boy attended robotics and coding classes during the lockdown, and later he took part in a competition where he secured the first place for developing a contactless liquid dispenser. A 10-year-old could think of a product that will help avoiding unnecessary touching of tap surfaces to dispense liquids. I'm sure we have that potential in us to be innovative and creative during these difficult times. I'm sure the members of the Microbiological Society will come up with innovative ideas to bring out the best in us. You have an enthusiastic and ever-supporting faculty in the Department of Microbiology. I wish the society all the very best for the academic year. I thank Dr. Poppy Datta and the office bearers for inviting me for this program. Have a wonderful evening. Thank you.
Thank you so much, sir, for your encouraging words. Unfortunately, Dr. Rabai, Vice Principal of Postgraduate Section, could not join us today due to some last-minute changes in his schedule. Nonetheless, now I hand over the platform to Larissa the General Secretary, and Amanda, the Joint Secretary, to introduce the association and office bearers.
Thank you, Madiha. Good evening, everyone. My name is Larissa Gomes, and as the General Secretary of the Microbiological Society, I would like to brief you about the Student Body Committee of the Microbiological Society, which is a part of the PG Department of Microbiology of St. Joseph's College. The Microbiological Society functions through the inputs of us, the students, by the students, and for the students. The Society enables learning outside the classroom, which provides rich experiences, opportunities, challenges, and responsibilities. This enables a holistic development of the students into active citizens and future stewards of the environment. The faculty coordinator of the Society is Dr. Poppy Datta. And now, I present to you the office bearers of the Society for the academic year 2020 to 2021. From the PG second year, we have Jennifer, who is the president of the Society, Larissa, myself, as the General Secretary, and then we have Aisha as the editor and Samson as the treasurer of our Society. That's about it from me now. I now request Amanda, our Joint Secretary, to take over and introduce the other office bearers.
From the PG first year, we have Caldera Paul D'Souza as Vice President, myself Amanda Taffy as Joint Secretary, Shivani Maiker as Co-Treasurer, Sanshil Matthew as Co-Editor, and Anusha Ghosh as the PR and Media Coordinator. As the newly elected members, we are looking forward to work with the current members, putting forth our ideas, leading to great synergy. Thank you. I now hand over the platform back to Madiha.
Thank you, Larissa and Amanda. Wishing you all the very best. I now request Aisha, our editor, to give us the association report for the academic year 2019-2020.
Thank you, Madiha. Good evening, everyone. This is Aisha, editor of Microbiological Society. I would like to summarize the association events of the last year. Our association has had quite an eventful academic year. We have organized multiple student development workshops and lectures. We had Dr. Ipsa Jain, postdoctoral fellow at Institute of Stem Cell Biology and Regenerative Medicine, Bangalore, conduct a workshop on approaching career goals and conducting dissertation research. Dr. Susan Phillip, Assistant Professor of the Department of Biotechnology, St. Joseph's College, Bangalore, delivered a seminar on effective resume building. We also had a lecture on scientific communication and medical writing by Dr. Namita A. Kumar, Founder-Director of Aspire Career Paths and scientific writer at Strand Life Sciences, Bangalore. Furthermore, Microbiological Society has hosted several academic lectures by esteemed scientists over the course of the past year. We've had Dr. Dipshika Chakravorty, Professor of the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore, talk about host evasion and vacuole hijacking by Salmonella and the need for antibiotic stewardship. Dr. Colin Jamora, Associate Professor and Laboratory Director, IFOM INSTEM, National Center for Biological Sciences, Bangalore, led an interactive discourse on wound healing and its related diseases and how this knowledge could be applied to potentially alleviate certain pathological conditions. Dr. Filippo Prisci, lecturer of the School of Life Sciences, University of Essex, UK, delivered a lecture on small cell lung cancer and a possible adjuvant therapy for the same. These lectures have been a huge success with students as they provide insight into current goings-on in the field of research as well as the opportunity to interact with scientists. Last September, we hosted our annual Intracollegiate Fest, Synergy, which included an exciting lineup of events like quiz, poster presentation, treasure hunt, just a minute, and Petri art, to name a few, with impressive participation from colleges all over Bangalore. We hope that this academic year will be even more fruitful than the last. Thank you. I now hand the platform back to Madiha.
Thank you, Aisha. I now call upon Jennifer to deliver the presidential address and give us a brief plan of the events for this academic year.
Thank you, Madiha. Good evening, everyone. It gives me great pleasure to welcome you to the inauguration of Microbiological Society for the academic year 2020-2021. I, Jennifer Albert, a second-year student of the department, with great honor hold the position of the president. I would like to thank the Almighty, the college, the management, and the professors of the department for this opportunity and for their unwavering and gracious support. I would especially like to thank Dr. Sen for accepting our invitation. It is a great pleasure to have you here with us today. I would also like to thank our members and the audience for your presence. I take this opportunity to congratulate my fellow elected officers. I look forward to working together and learning to steward our extraordinary fellow association members. We face a challenging and different environment today, and all of us have been inundated with what's happening around the world on COVID-19. While it has created new learning opportunities for the lot of us, it still bears the time of precariousness and devastation. Nevertheless, I'm particularly delighted to be entering this phase with a supportive team and a chance to adapt, show resilience, and explore fresh avenues despite the many restrictions in the time of this pandemic, much like our microbial counterparts. With that in mind, I'd like to project our plans for the year, which involve an array of interesting activities and events to keep our members and students engaged throughout. To start, we will organize a series of monthly lectures wherein we arrange guest talks from eminent scientists, academicians, and industrialists that they may share with us their expertise and in good turn expose our students to a range of latest research and developments. Next, we hope to organize a gamut of interactive events and competitions like Petri plate art, collage, poster presentation, pick to word, among others, either as an online or offline event, part of an annual fest. In December 2020, we anticipate releasing our annual newsletter, providing an opportunity for our students to publish their entries on varied topics. Finally, and importantly, we expect to have an increased social media presence, engaging our followers with a host of polls and interactive posts, cartoon illustration contests, and such like. That concludes our brief proposal for the year. We have great opportunities and some challenges, but with conviction, we can make it a great year. Thank you. That's all from me. Back to you, Madiha.
Thank you, Jennifer. I now request our Assistant Professor and PG Coordinator, Dr. Poppy Datta, to introduce our chief guest for the day.
Namaskara. It is indeed my pleasure to introduce Dr. Shubhojit Sen. As a scientist and an educator, Shubhojit is interested in the epigenetic query of evolution and environmental triggers that pushes normal cells to become cancerous. Being a triple gold medalist during his academic career, he obtained a PhD in molecular biology for his study on DNA repair and recombination at TIFR in 2001. Dr. Shubhojit initiated his work in the field of epigenetics at the National Cancer Institute, NIH, USA in 2002, followed by his work in tumor biology at Johns Hopkins Medical Institute, where he translated some of his fundamental work into the molecular discovery of epigenetic pathways that drive gene silencing in cancer. He returned to India in 2013 as a DBT Fellow at the UMDAE Center for Excellence in Basic Sciences, Mumbai, where he heads the Laboratory of Epigenetics, which focuses on understanding stress-induced triggers in cells that eventually drive epigenetic disorders. He has been felicitated with several prizes, including the J.N. Petit Memorial Award, the Gita Lopo Memorial Award, the Fogati International Fellowship Award, the prestigious Ramalinga Swamy Fellowship Award by the Government of India, and most recently, the Anil Sadgopal Award by HBCSC TIFR for his science popularization. Dr. Sen's recent work has been published in journals like Cancer Cell, Plant Molecular Biology Reporter, Protease Journal, to mention a few. His previous publications include journals such as Science, Molecular Cell, nature structural and molecular biology, BMC genomics, etc. Although fundamental in nature, till date, his work has close to 2,400 citations in peer-reviewed journals. Dr. Sen is also a member of the ISRC, that is Indian Scientists' Response to COVID-19, which does science outreach work and bust information during the pandemic. With this introduction, I invite Dr. Subhojit Sen to deliver his talk on tracking stress-induced epigenetic changes from humans' cancer to Chlamydomonas. Over to you, sir.
Thank you very much. I thank the organizers for inviting me to this occasion, and I'm happy to be here. I'm very happy that you guys are already doing so many activities. Particularly, thanks to Drs. Melvin, Dr. Beatrice, Madiha, and of course Jennifer, who got in touch with me to bring me to this occasion. So today I'm going to talk to you a little bit about some of the work that we have been doing, some of my previous work, which is already published, and I'll touch upon some of the paths that we are kind of going ahead with. And the broad theme of my talk is to shuttle between these two model systems, which is essentially humans, which is us, and Chlamydomonas, which is a green algae. So many people get surprised when I talk about these, as particularly human cancers. What has that got to do with green algae, right? So I'll just give you a quick. I mean, you've already heard my journey so far. So I got my PhD in TIFR, and I was fellows at NIH in Bethesda in USA, and followed that by Johns Hopkins. And currently a Ramalingaswamy fellow at Center for Excellence in Basic Sciences. And when I joined, you can see this little hutment-like things that you see, not a fancy new buildings that you guys have. So this is where one of these rooms was where my lab started in the institute when I first started. So, and these three beautiful ladies are the main torch bearers of the work that I'm going to present to you today. This is Snehal, and that's Patricia, and this is Pooja. Okay, and there are several people who are who have been part of the journey, and I'll mention their names as we go along. In 2019, which is last year, we moved into this new building, and that's where my lab is currently, in the new building, and all of this is inside the Mumbai University campus. We are a DAE institute, and we have similarly, UGPG programs in our institute as well, and this is centrally funded. So to begin with today's talk, because today we are in this COVID-19 pandemic situation, and people, we wonder what is the relevance to everything that we're talking about to what is happening currently as well. So I thought, why not we talk about that a little bit before we get on to the meat of the talk. So today, this is something that I picked up from one of the recent publications, which was in June, if I'm not mistaken. So by now, about more than 107,000 whole genome sequences of SARS-CoV-2 are available in public funded databases. Now, this particular paper, which was in June, had interesting analysis of the kind of mutations that are accumulating in SARS-CoV-2, right? And what they have shown is that there are plenty of mutations. Of course, in spite of so many mutations, let me draw caution that the rate of mutation is not as high as it would be, for example, in HIV or some of the other retroviruses that we know about. So, this is being an RNA virus, it copies from RNA. And so, therefore, the mutation rate is not as high as the HIV viruses, okay, that retroviral family. Now, what was interesting to me in this particular paper was this figure. And this is something that I want to ask the students here, and probably a poll maybe that somebody might shoot out to you soon. So my question to the students here who are attending this talk is that check out the numbers. So essentially what this shows is that how many times have they found the mutation from A turning to a G, or G turning to a T, or T turning to a C, and vice versa, all these different combinations possible, right? So why don't you look at the image and try to figure out what is the one mutational change that really, really stands out for SARS-CoV-2? And why don't you poll on that? And let's see what the students come up with. Can I request somebody to post the poll, please?
I'm having a slight difficulty in posting the poll. It's
All
not
right.
going through.
That's fine. Then if anybody can unmute their microphone and tell me from this diagram what they think is one of the mutations that really stands out compared to all the other mutations.
Sir, I think it's C to T because it has 1670.
Yeah, right. Exactly. This is not rocket science to anybody. Anybody who looks at this figure will find that that particular mutation, the C2 mutation, has a huge difference compared to any of the other mutations, right? So almost even, say about threefold different to the next biggest number, or even 10 times different than some of the other numbers, right? So there's something special with this mutation. So let's think about that a little bit. Okay. What I wanted to kind of translate from the title of the talk is something that stems from the understanding of this factor, the mutation that we are talking about. And so essentially the question that we are trying to understand today is, do epigenetic changes directly affect genetic mutations and therefore our path, our evolutionary destiny, or our evolutionary path that we are going to go through, right? And what I'm going to tell you now, in my next few slides, is how the C to T transition can happen and why the C2 transition is at such a high rate. Okay. Now this is a human gene I picked up. I don't remember the name of the gene, but I picked this gene up, and what I did was we have highlighted in this case on the left hand side, you see I've highlighted all the CG dinucleotides. On the right hand side, I've highlighted all the GC dinucleotides. So they are just flip flops of each other, right? CG. This is 5' to 3'. So 5' is CG, and so it's called a CPG, or 5' to 3' a GPC. And what you'll notice very distinctly is that near the promoter, which is the start site, so you can, this is the ORF start, and so the start site is here, somewhere here where the upstream, translated region is. So near the start site, where the promoter is, there's a huge clustering of CG, right? And then if you go away from the start site, you look at the rest of the genome, the CG clustering disappears. And that's not true for these other dinucleotides, right? And if you do this analysis for any other dinucleotide, you will see this. So something is special about CPGs, okay? So there's something interesting going on about CPGs. So what could it be? So now I'm jumping the gun and I'm going into epigenetics a little bit. And this is sort of the normal case of epigenetics in a normal cell, where tumor suppressor genes, which are our warriors and keep the tumors at bay, are essentially genes that will control the growth of tumors, right? So if they are being expressed, very likely, so this shows that the gene is being expressed, very likely that you may not end up with a tumor, okay? So these are surveillance genes and checkpoint genes and many such genes that fall under this category. Now, what has been shown in this figure is the epigenetic signatures of the nucleosome density, arrangement of nucleosomes at the promoter of this particular gene versus the rest of the genome, okay? What you can see clearly is that the promoter of these genes is largely open, or quote unquote euchromatic, and the rest of the genome is closed or has a closed conformation, or quote unquote heterochromatic. Okay, so the promoter region also has these lollipops that you see, and these lollipops are essentially the same CPGs that you saw in this previous slide, right? And what you see is that the white lollipops indicate that these Cs are unmethylated at the promoter, while at the rest of the genome, most often these CPGs will be methylated. Okay, so and notice I'm highlighting that the CPGs are being methylated. It's not the other types of Cs, it's the CPGs that are being methylated. So there are several chromatin factors that bring about these processes. This remodeling proteins are essentially proteins that rearrange the chromatin, the architecture of the chromatin. There are machineries that mark chromatin, and chromatin, in this case, the histone modifiers will mark the histones that form the nucleosomes, and DNA methylation machinery will put these lollipops. These black lollipops are put in by the DNA methylation machinery. So this is the cytosine, right? So now, if you imagine methylation of the cytosine, it gets methylated at the 5th position, which is right here. So if you methylate the cytosine, essentially you end up with 5-methyl cytosine at the CPG. But what is interesting is that there are these enzymatic processes that are rampantly occurring in cells. For example, oxidative reactions or deamination reactions. And if oxidative deamination takes place of this particular methyl cytosine, you lose this amino group because of the deamination process, and you end up with this molecule, which is essentially a thymine. So simply by enzymatic deamination of a methylated cytosine, I can end up with a mutation at the same place, which was originally a C, end up with a T. So this is the reason why you see a lot of CTT transitions, even in viral genomes and in across evolution. Okay, so there is something very interesting that there must be some evolutionary role of methylation, at least in this directed mutagenesis process, right? So let's try and understand as to how this is important in disease manifestation and how this might be, how this is, why we need to understand this process particularly to evaluate diseases like cancer. All right, so let me start this argument by putting down this slide, which I call the Big Bang of life, right? So this, if you look at the x-axis, which is on both sides, so from 0, you're basically looking at the timeline of evolution till today, present day today. And this is essentially to give you the species evolution, so the tree of life, so to speak, right? And what you can see is that there are several families, and you can see clearly, like this is your unicellular bacteria archaea. Then you have the simpler eukaryotes and the complex eukaryotes from plants. You have the fungi, the simpler and the complex fungi, and then of course the rest of the tree that finally translates to all the way to humans, right? So clearly, all the organisms that are present today on the surface of this bubble are existing today, and the organisms, for example, here you can see the dinosaur extinction that happened, right? The mass extinction of dinosaurs. Each of these lines, as you can see, are an extinction event, right? All right. So how to think about this tree in a more in a more realistic fashion is to think about humans appearing, right? If you think about the evolution of Earth to be compressed within 1 year, that Earth was born on the first stroke of midnight of 1st January, then when did humans come into being? Humans came into being somewhere around like 12th December or 15th December, and the first civilizations came around only in 5 minutes to midnight. So you can see that we have all the science development, all the development in modern understanding of how things work have really happened in a very, very short time. Now, how can we understand, how can we relate to diseases, how can we learn from all of the previous history that we have in evolution, and how can we extract from that information and learn about humans and apply these things to humans, right? So that is the sort of the quintessential question that everybody wants to ask. So one of the things that I'm going to put forth is that we do need new model systems for biology, because obviously I can't do every experiment in humans, right? So I have to figure out new model systems in biology. And what I've highlighted in these slides now by these circles are several simple model systems that many people work on, and particularly these ones because I have worked on them through my career, through my 25 years of research career. And today we are focusing on these two model systems, which is humans and Chlamydomonas in our laboratory, okay? So what does evolution teach us, right? We know that there is this common ancestor. We are all born from this LUCA, which is the last universal common ancestor. So there is a common tree or common methodology to the madness of understanding biology, right? And particularly if you're looking at processes that are conserved across evolution, for example, DNA replication. If you look at DNA replication, I can study DNA replication even in bacteria, and that mechanism will be applicable even to humans, right? So if I look at a DNA replication inhibitor, and I've discovered that DNA replication inhibitor is working in bacteria, that same DNA replication inhibitor will also work in humans with some modifications here and there, but let's not go into the details. But the understanding is that this evolutionary relationship helps us look at complicated processes in a much simpler fashion, right? So essentially, this is what I called as reducing the problem. So how do we reduce a problem like cancer or diseases like cancer, for example? So let's understand quickly what cancer is. I'm sure most of you are already familiar with what is the etiology of cancer, but this is the modern sort of one slide understanding of cancer. So normal cells become cancerous. How? So there are genes that maintain homeostasis in cells, and this is what is maintaining homeostasis looks like. There are growth promoting pathways and there are suppressor pathways, and the balance of these pathways is what maintains homeostasis, right? So I would imagine them, if you're driving a car, you can imagine these genes as some of them being the accelerator genes and some of them being the brakes of the system. So ideally, you don't want to overdo one or the other, because otherwise the car will crash, right? So you essentially think about this paradigm now in the central dogma. In the central dogma, we know that in normal cells, when a normal gene is being translated, we end up with a functional protein. And this functional protein eventually gives rise to the phenotype, right? And we know that environmental factors like carcinogens can mutate DNA and thereby lead to non-functional proteins and thereby lead to misfunction of these genes or mutation of these genes, lead to non-functional proteins or inactivation of these genes, and therefore lead to mutated phenotypes or mutated genes, which result in cancer. Right? This happens largely by either gain of functions of accelerator genes or loss of function of breaks or tumor suppressor genes. And what I want to highlight is particularly in this mutational hypothesis of cancer, loss of function plays a major role because it is seen that many genes undergo loss of function in cancers. And there's a different evolutionary argument for that as well, but let's not go into that detail. The essentially loss of function is commonly seen in many cancers. Multiple loss of function happen, and a few gain of functions, which are your oncogenes, happen in cancer. All right, so there is this little bit of a twist in this paradigm, which I want to introduce now. The twist is that the insult that results in cancer and the injury, which is cancer itself, are not usually coincident. And this has become a large problem in cancer because, we know that smoking causes cancer. We have known that for 50 years now. But many people continue smoking even today because, you don't pick up a cigarette and end up with cancer tomorrow. So that you don't associate the insult and the injury together. And because of that factor, many people still don't appreciate how smoking can, drive cancer. Now, why is it that there is a separation between the insult and injury? And let me give this example in a little more in a different fashion. So this is, when we go from zygote to old age, right, all the way. And we know several non-communicable diseases now that follow this pattern. We know that malnutrition, early life malnutrition, can result in what is obesity later in humans. And fluctuations in diet are highly correlated to neurological disorders. So how is it that the insult and the injury are separated by several years, while we know that, for example, in the human body, by and large, within a span of 2 years, you are completely a new organism because all cells have recycled, all the old cells have died, and practically within 2 years, you are practically a new organism. Like read about the concept of ship of Theseus, right? For example, it's an old Greek concept. Essentially, how does the body remember what was this early insult, and that results in disease later on in life? So what predisposes us to disease? So this is what gives us the hypothesis, an epigenetic hypothesis, which is that there must be a transmissible cellular memory which is being transmitted across generations as the cells divide. There's some memory in the cells that is transmitting across generations and finally predisposing us to these non-communicable diseases, right? And what is now known is that this memory can also be transgenerational, okay? Intergenerational or transgenerational. Now, this is where our work is focused into, where we shuttle between these two model systems. And today I'll first talk about humans and then talk a little bit about Chlamydomonas in that respect. So coming back to the mutational hypothesis of cancer, right? How else can we think about cancer now? How can we think of it in an epigenetic fashion? So we can think about the environment playing a major role here, because all of these factors that I just talked to you about has an environmental insult involved in the process, right? So if you think about environmental insult, if environmental insult is somehow able to obstruct the DNA of information from being read, then essentially you are going to lose this pathway and your protein is never going to be synthesized. Even though you have a wild type gene, you will never be able to synthesize the protein, right? So you essentially end up with a loss of function, and that loss of function will result in the loss of the function of tumor suppressor genes, which will eventually lead to imbalance in the system, right? And this imbalance leads to a gain of function of oncogenes because mutations can now occur without check, and that mutated gene can eventually give rise to cancer. So this is kind of what I call the epigenetic hypothesis of cancer. Okay, remember, this is a hypothesis. This is something that many people have proposed, and we are also kind of trying to work in our work is kind of indicated many of the pieces of the puzzle that give rise to, that is put credence to this particular theory. Okay, so let me talk about that part. So question is how, if this is epigenetic, then one must therefore expect that there must be a large overlap between the cancer genome and the cancer epigenome. Okay, and I'm not going to go into the details of these two comparisons today, but let me give you a quick some data about how this goes about. So I know that epigenetics is not something that is taught at many undergraduate levels and postgraduate levels, and many people are still confused about definitions of epigenetics. So that's why I'm kind of, people confuse transcription with epigenetics as well. So what is epigenetics, right? So let me clarify that by this one line definition, which is essentially heritable changes in gene expression. Very important. Heritable changes in gene expression that are mechanistically rooted in chromatin changes without mutating the primary sequence of DNA. So transcription also has half of this common, which is essentially if you can have transcription with control, you are without mutating the primary sequence of DNA, you can end up with transcription control, right? But that may not be heritable, or even if it is heritable, it may not be through changes in chromatin sequence or changes in chromatin architecture or chromatin mechanisms, right? So when you stitch all these three together, that is what gives rise to epigenetic changes. Okay, this imparts normal development, differentiation. Essentially, it boils down to how our DNA in our systems is packaged, in our cells is packaged. And the cancer epigenome, if you think about it, is therefore the abnormal manner in which cancer cell DNA can be packaged, okay, which leads to heritable patterns of gene expression. All right. So how is this studied? And this is one of the most common ways this problem is addressed. And you have used and worked on this problem quite a bit. So so essentially this is just showing an yeast cell, but the same can be done with clammy, can be done with human cells, you name it. Essentially you generate, you want to study this substrate, which is the chromatin. You generate pieces of chromatin, which is essentially a chromatin substrate, and do a procedure called chromatin immunoprecipitation. Okay, let me quickly tell you that in Chlamydomonas, when we started our work in Chlamydomonas, there was no way to generate this particular substrate. So we actually designed a 5-minute assay through which is a single tube 5-minute assay, which could develop sort of chromatin, these type of chromatin ladders. These are called chromatin ladders, which are essentially repetitive units of chromatin, which can be digested by an endonuclease, and you can generate these ladders. Okay, so we showed that we generated two types of methods that could do this in Chlamydomonas and generate micrococcal nuclease or endonuclease derived chromatin ladders from intact algal cells. Okay, now this is important because most protocols require you to extract the nuclei first and then extract the chromatin from the extracted nuclei. Okay, whereas our procedure goes straight from intacted cells to the chromatin. So that is one interesting aspect, and I'll come to later as to why that is important. But what do we do with this extracted chromatin template now? So this is the schematic of chromatin immunoprecipitation. I'm sure all of you know what immunoprecipitation is. So essentially, you're using an antibody to pull down a specific mark. We know there are histone modifications like acetylation or simulation or methylation. There are many, and there are antibodies that are available that are specific to each of these marks. Okay, I'll come to the details a little bit later. But essentially, if you bind these antibodies to protein A magnetic or in agarose beads or magnetic beads, these are coated with protein A. The antibody can bind to these beads, and now when you throw these chromatin templates on these beads, the chromatin will be recognized specifically for that mark that you have looked for, and the chromatin can be purified, right? That particular chromatin can be enriched. Okay, so now once I've enriched this chromatin, because I can just centrifuge the beads and just enrich this chromatin from the solution, I can elute this particular antigen-antibody complex by dissociating these interactions. And once I elute this, I can remove the beads and digest all the protein in this complex. I can remove the beads and digest the protein, and all I'm now left with is the pieces of DNA that were originally coated on the specific histones that carried that epigenetic mark, right? Now, because I can do that, I'm left with this DNA, and I can query as to which mark is existing on which part of the genome. Okay, so this is where the genomics begin. And so we start by candidate gene analysis. Okay, what I've shown you is that because we enrich this in very small quantity, we can amplify them by using PCR, right? So I'm showing you a technique of how to use PCR to query where the epigenetic marks are enriched. So in this example, you can imagine thinking of, again, of the gene that I'd given you before. You have the promoter, which is essentially the business end of the gene, where the regulation occurs, and then the rest of the gene, right? So there are nucleosomes present all over the gene. The question is, what type of marks are enriched with the promoter versus the rest of the genome, right? So let's think about it in a binary fashion, although these are not exactly binaries. These are very tunable epigenetic reactions, but let's think of it in binaries for simplicity of understanding. Let's look at an expressed gene and a silenced gene, where the gene is either expressed or no mRNA is being produced when it's silenced, okay? So if you think about it, let's design PCRs across this entire gene, and we can walk, essentially using the PCR, we are walking across the gene and asking which of these nucleosomes is enriched with the original mark that we queried for using the antibody. Okay. So now if I used a KH3K4 antibody, what I noticed is that these nucleosomes near the promoter were enriched because I saw higher PCR signal there. And for an H3K27, the same H3 protein, but a different lysine that is marked with methylation actually gets enriched in different parts of the gene when it is expressed, but not at the promoter. Okay. And it becomes vice versa. This entire phenotype is reversed. This epigenetic phenotype is reversed when you look at a silenced. That is, the active mark, which is H3K4 trimethyl, is not found very often there, whereas the repressive mark is now far enriched at the promoter than compared to the rest of the genes, rest of the genome, right? So this is how a typical reaction would look, and this is exactly the experiment that I just detailed to you about. And you can see that the promoter is somewhere. I mean, this is where the ORF is, and the transcription start somewhere is in this region here, right? So what we noticed, and in this example, I queried two types of epigenetic marks. One is known as a variant histone. This is H2AZ or H2AZ, which is a variant of the histone H2A, which makes up chromatin. And there's another mark which I queried, which is H3K4 trimethyl, okay, which is the active mark, okay? Now we notice that the active mark as well as the H3K27 has two distinct peaks around the transcription start site, right? And as I walk along the promoter, the region where the transcription start site is, I see these two peaks that are enriched, both for the H2AZ as well as for the H3K27, right? So this is how a typical reaction is done. And this is what you're seeing here is a PCR gel for the same reactions. And these are your controls, okay? I'm not going into the details of the reaction, but these are your controls. Essentially, what this observation tells you that there is a coincidence of these active marks with the H2AZ, and that hints to the biology of the system. Let's talk about the biology of the system. Okay, so this is sort of, I have now compiled a lot of things in epigenetics in one slide, so it is going to go really fast in this slide. Okay, but I'm going to try and take you with me in this. So this is what I showed you before, which is the scenario of normal cells. That in normal cells, most of the promoters are unmethylated and therefore expressed, and rest of the genome is methylated and therefore repressed. Okay, these I'm talking about CPGs. Remember, I'm still talking about CPGs now. But if you think about it, all of these are enzymatic reactions, right? So therefore, they are sensitive to the environment. They are sensitive to oxidative stress. They're sensitive to all kinds of stress, to different kinds of metal ion stress. They are sensitive to the diet that you eat. They are sensitive to the environment you live in, the food that you eat, all kinds of things. Right? So what happens, what we know is that when you look at the same phenotypes in a cancer cell, the phenotype is completely switched around, right? One, you'll see that the CPG are hypermethylated at the promoters, and the rest of the genome, CPGs, are hypomethylated. So the level goes down in the rest of the genome. And the second thing is that the promoters, these tumor suppressor genes that we're talking about, are now more closed conformation or heterochromatized than compared to the rest of the genome, which is seemingly more euchromatized than compared to a normal cell. Okay, so this dichotomy seems to pop up quite often when we look at normal versus cancer. The question is, how do we get from here to here? Right? These are the endpoints of the reaction. We already get in a cancer cell, we already observe these things. But how do we get from here to here? Right? What is this process? And that is the first question. That is the question that I started asking when I got into the field. This was way back in 2010 when I started asking these fundamental questions. Okay, so the way we started asking these questions is we started looking at a couple of histone modifiers and DNA methylation machinery. After scanning a bunch of epigenetic proteins, we started asking these questions. So today's talk, I'm going to focus on these three pathways. Okay, two of them are histone modification pathways. These two modify. This one puts the H3K27 trimethyl mark, and this one puts is a histone deacetylase, which basically removes the acetyl mark. Essentially, both of these marks, as well as this mark, all of these are repressive marks. Okay, which means that they are responsible or they are highly correlated with gene silencing or gene repression. Okay, DNMT3 and 3B and DNMT1, these are the enzymes that are performing the DNA methyl transferase activity. Okay, so essentially these three pathways are independent pathways as far as the enzymatic activities are concerned. So let's understand that how this works. So what is known in epigenetics is that if you look at these three pathways, they are arranged. I've particularly arranged them in this fashion because this one is the most repressive pathway. Then comes this one, and then comes this. So repressive quotient of this is very high. The DNA methylation is very high. Okay, so polycomb is somewhere in between, and histone deacetylase is relatively low in its repressive quotient. And the plasticity quotient, which means how I can undo these interactions, right? How reversible these effects are, is exactly reverse of the repressive quotient. Okay, and this has to do with how the epigenetics are heritable. So these marks, I remember I told you it's heritable changes in gene expression. So how these marks can be heritable across cell divisions is reflected in the mechanism of this repressive quotient. That the more the repressive it is, the less plastic it is. The least repressive it is, the more plastic it is. Okay, so remember these dichotomies of that expression. Of course, these are all on a scale of on many shades of gray. These are not just binaries. Okay, none of these are binaries. So let's think about it as to what is happening in a human cell in these proteins. Okay, so now what I'm doing is I'm showing an experiment which we had performed. This is in a stem cell type of model. And what we are seeing is we have extracted these proteins. So here we have the DNMT. This is the 31, which is the histone deacetylase. Again, another DNMT. And these three are part of the histone, the polycomb machinery that I'm talking about, which makes up the histone trimethylation, K27 trimethylation machinery. Now, what you're seeing here is that I have taken these proteins and we have centrifuged these proteins, extracted these proteins sort of from the native nucleus freshly, and we have immediately centrifuged these proteins through a sucrose gradient. Okay, what the sucrose gradient essentially does is based on the radius of the molecule, or what is known as the Stokes radius, the perceived radius in solution in fluid dynamics, it's called the Stokes radius. Based on the Stokes radius of the particular molecule, this molecule, if it is larger Stokes radius, it will be more buoyant because it will face more resistance as it is going down into the solution. And therefore, if you think about it, as increasing densities, the Stokes radius is going to be decided by the density of the complex, okay? The density and the packing density of the complex. So the more deeper this protein goes into the particular gradient, the more denser or the more compact this complex is, okay? What we notice is that in normal cells, when we take normal stem cells and we look at untreated normal stem cells, most of these proteins, and this is a Western blot of the eluted sucrose gradient, you see that most of these antibodies are detecting these proteins and they are in the upper region of the gradient. And they are not really deep into the gradient. Okay, but then we treated these cells with hydrogen peroxide. Okay, and let's see what happens. Before that, let me just emphasize that these three pathways essentially do gene repression independently. Okay, they are doing independently as three different complexes. But the moment we treat them with hydrogen peroxide, which is basically a form of ROS or reactive oxygen species stress to the cells, look what happens dramatically within 30 minutes. And this is mammalian cells. So within 30 minutes, you see that, and this human cells, not just mammalian, these human cells, within 30 minutes, you see that all these proteins are shifting much deeper into the gradient. Okay, so what exactly is going on? So we hypothesized at the time that maybe these three proteins are now all coming together, forming a larger complex, forming a much more larger, say about a 2 megadalton complex. And that is what is probably initiating this process. Something is changing in the way these proteins interact. Now, these three proteins are interacting, not these proteins, they are multiple proteins, but these three pathways are interacting and they are coming together, forming this complex. Is that true or not, right? So the way we ask this question is by co-immunoprecipitation, again, which is another sort of usual classical way of asking the same question. And sure enough, what we noticed is that if you look at, these are the same fractions, 1, 2, 3, 4, 5, the purified fractions, and you notice that between this set and this set, if you look at this set and this set, the third fraction is much more enriched with these interactions than compared to the third fraction here, which is essentially this part, right? So these are actually interacting with each other. The co-immunoprecipitation proves that they are interacting with each other, and that is how they are actually one big megadalton complex that has formed. So question therefore is that ROS is initiating this megadalton complex formation. These are all epigenetic proteins. Where are they going in the genome? Okay, what are they doing? Okay, so this is a whole genome analysis of the same thing. What I'm showing you is chromosome 21, and the red line is essentially the line of normalization. So we have normalized against the untreated control. So these are each of these black lines. So this is the entire chromosome 21 sequence. Of course, it's very dense. So let's not think about it. Let me just tell you what the graph indicates. And this, every dot here is a signal, right? And the higher the dot means more the signal once it is ROS treated, And the lower the dot means there is a loss of signal upon Ross treatment. Okay, so what you see is that there is a much gain of signal in this part of the chromosome upon Ross treatment, and loss of signal in this part of the chromosome, which is very unique for this chromosome. Okay, in other chromosomes we saw a large distribution. So we started asking as to why there is such a increase in, and this is true for all the epigenetic proteins that we have that have taken you through so far, right? Not true for H3, which is a histone, which is normalized across the entire genome, right? So you can see that H3 does not change, which is there in every nucleosome, whereas only it is in this arm this increase. There was a gain of signal. What we noticed is that in this arm of the chromosome there was higher transcription signal, and in this arm of the chromosome there was a higher percentage of GC bias. Okay, so the concentration of GC was higher in this particular region of the chromosome. So of course, when we looked at all the genes that are getting methylated, not methylated, all the genes that are getting silenced by these processes, there was a huge overlap of all the genes that had all the proteins. Okay, so let me cut the story short. Of course, this is a lot of many years of work, and essentially this was published way back in 2011 in Cancer Cell, and essentially that oxidative damage targets these complexes containing these proteins to promote a CPG island. Okay, so what do they do at these islands, CPG islands? Those clusters of CPGs that I showed you in the beginning of the talk, they are called CPG islands. Okay, so in the stem cell model, what we noticed is that when upon treatment, some of the tumor suppressor genes, so these are 3 tumor suppressor genes which have different expression levels. The lower the expression of tumor suppressor genes, the more the DNA methylation they accumulate. Okay, so this is upon treatment, there's more accumulation of DNA methylation, and this is just a measurement of DNA methylation. And again, this was done in not just in stem cell line models, but also in animal models, where we showed that when animal models were subjected to inflammation-based Ross treatment, so this animal was infected with a particular bacteria, which was a colonic infection, and that colonic infection gave rise to inflammation of the gut. And that inflammation, so all of this is naturally occurring, right? So inflammation of the gut leads to Ross, and Ross is how, you can see in a sham, which is essentially the control. In the sham, nothing much is happening, but in the ETBF, which is basically the bacteroid treated samples, in those mice, you will see that there was dysplasia of the colonic epithelium, right? So this is kind of the first signs that something is going wrong with the tissue. From normal tissue, it is becoming dysplastic, or quote unquote phenotypically going down towards the cancer pathway. Okay, so this dysplasia was something that we had seen in animal models. So clearly, there's DNA methylation as well as phenotypic dysplasia moving towards cancers, right? So let's understand this inflammation model a little better. So so far we have come across this pathway, right? So we have demonstrated that these complexes come together, lead to DNA methylation, which leads to cancer. Okay, so of course I'm not showing you a bunch of data. I'm showing you only highlights of the data that we have produced. And now the question was to look at it in stem cells as to what is it that is there in stem cells, because remember I told you that this memory is being transmitted from generation to one cellular generation to another, and so we are remembering this abnormal memory of insult, right? So what is in stem cell that is special about stem cells that is allowing them to remember this insult, right? So we thought about it a lot, and there was this interesting paradigm that we came across in stem cell biology. Now in stem cells, there are these 2 marks. This is the H3K4 trimethyl mark, which is the active mark. This is the enzyme that puts it. And this is the K27 mark, that is the inactive mark, which is input by polycomb. Okay, there are other inactive marks as well. But in stem cells, and all these marks are put by a combination of these 3 machineries, which are your writers, which put the mark, the remodelers, which rearrange the chromatin, and your readers, which read these marks and then they know what to do next. They interact with each other all the time, okay, if you think about it. Now, in stem cells, these 2 marks, although each of them seem to be opposite in nature in terms of its functionality, these 2 marks coexist on the same region, okay? And this is what is called as bivalent chromatin, okay? So this is unique to stem cells, that many genes in stem cells have bivalent chromatin. What is bivalent chromatin? I'll show you in a genomic study, for example. If you look at this region of the genome, you will see that this region has both the K27 peak, K4 peak, as well as the K27 peak, okay? So that's why that region is bivalent. And you notice that this is the nanog gene, which is usually highly expressed in iPS cells as well as stem cells, right? So it maintains stemness of the population. Now, what was not known about these, remember that what is important to understand at a molecular level is how do these bivalencies play out at the molecular level, because you are talking about opposite marks. If both the marks are there on the same nucleosome, it might be read very differently, because readers have to read this and know what to do. They will be read very differently versus if these two marks are on adjacent nucleosomes, right? That will be read very differently. So the combinatorial mark versus the independent mark, even though they are both there on the same region, they might be there on adjacent nucleosomes, so they are not going to confuse each other. So question was, how are these marks placed? So that's the question we asked next. So where are these marks both present on the same nucleosome, or are they present on different nucleosomes? How did we ask this question? Is by going back to the chip design and modify it by what we call the sequential chip design. Okay. So what happens in the sequential chip? You have the primary antibody, you do exactly the same thing, and then whatever you pull down with the primary antibody, that substrate now has one of the marks, right? Now you take that substrate and you again pull down with a secondary antibody, and that secondary antibody is going to pull down from that subset what contains the second mark. So whatever you're able to pull down after 2 cycles of enrichment have both the marks, right? So this is what happens. So essentially the first mark will bind to the antibody, which we elute, and then once we remove this, the antibody is also dissociated. Now we add the second antibody, but the problem comes here. Now, once we add the second antibody, we have not been able to remove the first antibody, right? The first antibody is still there in the solution. But now when we add the second antibody and we want to do the reaction, we want to do the enrichment reaction, both these antibodies will go and bind to the particular nucleosome, right? So we will be confusing the data. So how do we get rid of this factor? The way we went about it was going back to the design and modify the design. In the original step, when we added the primary antibody, we cross-linked the primary antibody with a cross-linker and fixed it to the beads. Okay, once we fixed it to the beads, what happens is when we remove the beads, the primary antibody also gets washed away, and we remove the primary antibody completely. By completely removing the primary antibody, we are increasing our signal-to-noise ratio in the secondary antibody step. And then we perform the secondary chip analysis. Okay, so this is essentially an example of sequential chip. I'll quickly just draw your attention to this particular region. I don't know if it's clear to many people, but if you notice that this particular band here, this particular nucleosome, the yellow nucleosome, is more enriched than compared to this nucleosome, which is the blue nucleosome. They're right next to each other, very close to each other as far as the gene is concerned. This is the gene SFRP1, and these two nucleosomes, this one is bivalently marked, that has both the marks, and this one is monovalently marked, which has only the K27 mark. Okay, it's not enriched with both the marks. So this is the secondary IP that shows the enrichment profile. So we took this gene-wise analysis, we took this candidate gene analysis and made a genome analysis, full genome analysis. And this is what we got. Okay, I want you to focus only on this graph right now, because this is the essential sequential chip. The rest are all controls. Okay, so for every experiment, we have to have multiple controls to show that we are not going wrong. Otherwise, it's very difficult to analyze the genomic experiment. So this is the meat of the experiment. What we noticed is that these sequential chip signals, which have both the marks, are only enriched at the transcription start site. So all these, if you look at this side, all the entire stem cell genes are now arranged with respect to this transcription start sites. Okay, so this is the transcription start at 0, and then the gene is downstream and upstream of the gene. And what we notice is that the sequential chip signal is largely enriched exactly at the transcription start site, right around there. Okay, so this means that the bivalent nucleosome is very important transcriptionally. How? Okay, and what we noticed next is that if you looked at the methylation step, the CPG, remember I told you that CPGs are also enriched at many of these genes. There are some genes in the genome that don't contain CPGs. Okay, so we can have some genes that don't contain any CPG islands, and most of the other genes have either high density, medium density, or low density CPG islands. Okay, what we noticed is that when you classify the genes based on their density of CPG islands that they possess, and you look down at the sequential signal, we completely lose the sequential signal in genes that do not contain the CPG islands. Right? So that means the CPG islands are somehow responsible. The genetic signal is somehow responsible for maintaining the bivalency at these promoters. Okay. Now it throws up an interesting thing, that what is happening to the methylation status there? Because we know CPGs can get methylated in cancers, right? So when we looked at the methylation status of these genes, now again you have to focus on this particular graph here. The others are again controlled. This is H2AZ, this is K4 alone, and this is H3K27 trimethyl. Okay. What you notice is that when you look at the sequential chain, when I classified the genes based on the methylation status, so red is unmethylated genes and black is the hypermethylated genes. So when the genes are hypermethylated, those genes do not have a high bivalent signal. Those genes that are unmethylated will still have the bivalent signal. So this indicates that bivalency is opposed to DNA methylation status. Okay. 1 minute. I will have to come back here. All right. So yeah. So what we have learned from this example is that bivalency is going to oppose CpG DNA methylation. Somehow it is anti-DNA methylation. And also H2AZ, which I have not told you in detail, and many other people have also shown. So bivalency we have shown for the first time, but H2AZ, many people have shown that it is also opposed to DNA methylation. Okay, so both bivalency and H2AZ seem to be, at least by our analysis, seems to be the last barriers in stem cells to cancer DNA methylation, is what our hypothesis is tending towards. So this is something we are currently working on, and this is something that is a future line of query. Now the question is, how does all of this cancer phenotype that we have talked about, how does this relate to the age of the disease, age of a person, right? Because we know that cancer is also an age-related disease. And this is just a quick study that I'm showing you. What we have found is that in cancer, in a cancer model where we are looking at cancer as a stochastic occurrence, right? Cancers happen randomly or stochastically. And in those models, you see that each of these circles indicate a CpG at a particular gene. The black one indicates a methylated CpG. So we see that the CpG methylation is random, right, in cancers that are stochastic. But if you look at another form of age-related phenotype, which is senescence. So senescent cells accumulate in older people. And if you look at the senescent phenotype, these methylations are also always very, very programmed, which means that this is very predictable. You will be able to predict which gene will have which methylation. Okay, and that's the data. I'm not going into the details of the paper, but this was also published in 2018, which showed that there is a distinct difference in the way cancers can get methylated. Okay, so let me not go into the details, but I want to now ask the next question, which we are kind of asking the lab currently, which is, what is the evolutionary understanding of this process? What is the evolutionary conservation of this process? Okay, so for that, I want to show
Less than a billion years ago, life crossed a threshold.
Listen to this.
Single cells started to band together, eventually evolving into the riot of shapes and functions of multicellular life today. It's been thought that the evolution of multicellularity must have been a major genetic leap, but new research suggests that it wasn't such a difficult transition after all. Scientists looked at an organism that seems closest to making this leap. Choanoflagellates, while single-celled, resemble the specialized cells that line the channels of sponges, which are considered by many to be the most primitive living animal. A lab found 350 gene families in choanoflagellates, once thought to be exclusive to multicellular animals. The findings suggest that this single-celled organism is already equipped for multicellular life. A single cell's life is simple but limited. Like hermits, microbes need only be concerned with feeding themselves. In contrast, cells in a multicellular organism give up their independence. They take on specialized functions. They sacrifice their progeny for the greater good. Take vulvar algae, which comes in a spectrum of cellularity. A lab compared the biology and genetics of this diverse set of algae and learned that as cells banded together, existing genes took on new functions. Chlamydomonas is a single-celled alga that uses its centrioles for both swimming and reproduction, so it can't do both at the same time. Its multicellular relative, Volvox, however, can do both at once, because its cells have specialized, with some using the centriole for swimming and others using it for reproduction. Yeast is a fungus consisting of a single cell. One lab conducted a series of test tube experiments with yeast cultures to better understand how cells evolved, increasing complexity. The biggest cells, which were the ones that settled to the bottom of the flask, were allowed to survive and reproduce. Within 2 months, daughter cells stuck to their mothers and formed multicellular structures, dubbed snowflakes because of their shape. Over time, they got bigger and more complex. In this sped-up simulation of evolution, the yeast specialized cells. In large snowflakes, certain cells undergo a type of suicide, releasing the cells at the tip to start a new snowflake. The dying cell sacrifices its life so that the group can reproduce, a hallmark of multicellularity. The team conducted the same experiment with Chlamydomonas, and again, a kind of multicellularity was quick to appear. Scientists are still debating what drove simple cells to become more and more complex. But with these genetic comparisons and clever experiments, what was once thought as a major leap can now be seen as a series of small genetic steps that led to the wondrous diversity of multicellular life today.
So again, going back to this idea of this, how to think about multicellularity, and why Chlamydomonas, right? So this is one of the pitches that we understand why Chlamydomonas, because it sits at the cusp of multicellularity. So right here, beyond these is when multicellularity started evolving. And Chlamydomonas comes from that cusp, right? So this Volvocine algae group comes from that cusp. So it shares many of the genetic, our evolutionary historic remnants of the genetic programs that led to multicellularity. So why am I discussing this sudden diversification of multicellularity, right? This is where I'm going to go out on an arm and a leg and sort of state this hypothesis. But I think it's very important to think about cancer in a different manner. Okay, and this is how to think about it. So if you think about human cells are these aggregation of cells which codependent on each other, right? And cancer is when this phenotype becomes deregulated and you end up growing a mass of cells, right? And what is important that these kind of cells undergo a transition known as epithelial to mesenchymal transition. And this transition is reversible in normal cells, but in cancer cells, it becomes irreversible, right? And that is what leads to the original cell leaving the tumor tissue or the benign tissue and becoming metastasized, right? So essentially, if you think about this problem, what is going on is that our cancer is breaking the rules that govern multicellularity and therefore metastasizing, right? So if you think about it that way, does it therefore become more important to understand the rules that govern unicellularity and that transitions from unicellularity to multicellularity, right? So what is the rules that the cancer cells are thinking about or responding to when they are undergoing these benign to metastatic or cancer transitions, right? So this is how to a new way of thinking about cancer. And we believe that, at least, I mean, this is one of the queries that we are currently doing in the lab is there any epigenetic basis to it? And I cannot go into too many details of those assays because everything is now linked to how does the environment affect these processes, okay? So let me step into Chlamydomonas now, which is the model system going for the rest of the talk. And I've given you a picture of how the unicellular organism looks like. It actually has an eye spot, which is exactly the same protein that we humans have, rhodopsin. And then it has cilia, which is another factor that we humans share with Chlamydomonas. And then it has a chloroplast, which is there in plants, right? So it's kind of has, and it's of course a motile organism, so it can move around, and so it shares with both the kingdoms. It shares some similarities and shares tremendous similarity with humans, right? As you can obviously see. So of course that's a joke. So the question is to understand: can we derive from our human understanding of cancer models in epigenetics and look for evolutionary conservation, or what is the minimalistic conservation of this evolutionary pathway in epigenetic basis in Chlamydomonas that can help us learn new things about cancer? Okay, so let me, so what we know about Chlamydomonas, why Chlamydomonas is of course because it has, not only it has the CpG DNA methylation, it has all other DNA methylation machineries as well. And it also has some of these other genes that I talked to you about earlier, which is the polycomb complex as well as the HDAC. So it actually has all these three pathways operating inside the cell, inside these single cells. Okay, so what are these pathways doing? Of course, we don't know, we don't have any of these answers today, but we have some clues to where we are going to go and look for. Okay, so let's step into molecular epigenetics. So what I've been describing to you so far are a lot of molecular assays in epigenetics, but how is that important, right? Imagine now this woman shouting, help a thief, right? She sees the police coming and she's very happy that the police is going to catch the thief who's running away. The police is actually letting the thief in and helping the thief, right? So and she's like, why aren't you arresting the man, right? How aren't you arresting the thief? Police is like, commas, lady, commas. And you notice it's help a thief, which changes the meaning completely, right? Which changes the meaning. So essentially, the genome language, how you express the genes, how these network of genes are expressed, how these are understood by the cell is entirely dependent on which genes are being expressed, how much of it is being expressed. This is what I call as punctuation. Okay, so this is essentially the punctuation of DNA accessibility, and this is the way in which it can happen. And each of these is an epigenetic mechanism or an epigenetic barrier or punctuation to the gene expression. And you will notice that it's going from open conformation to closed conformation to finally this typical X type of chromosome, X type of shape that we see in our textbooks, right? So there's many things in between. So it is important to understand these punctuations. In fact, it is in the mechanism of this punctuation that lies the syntax and the grammar of genome language, which is the packaging. Okay, so there is a lot of strong evidence on this side and a lot of strong evidence on this side about epigenetic mechanisms, very little or scarce evidence in the middle. Okay, and there are different reasons for that, which I'm not going to discuss today, too much of it, but let me see how much I can carry forward in this direction. So to think about this now, as I just said, that there is enough evidence in many of these other types of understanding, and that is clearly because of the type of tools that are used to understand these epigenetic mechanisms. Okay, when you look at the chromosomal mechanisms, you use cell biology mechanisms. When you look at the chromatin mechanisms, the detailed the nucleosomal mechanisms, as I've shown you so far, we use molecular biology, right? And to understand these multiple steps, we use biophysics or biochemistry to understand. If you think about it, each of these techniques are not talking to each other, right? So essentially, we are looking at this old problem again. We are looking at the epigenetics from a very narrow point of view, and we don't know whether we are looking at the complete picture. So our aim, one of the aims in the lab is to generate assays that are more holistic in nature, that can talk to each other and be able to transition from one form to another, and we can link these different types of understanding to understand what the epigenetics is saying. Okay, so I'm going to present one such assay to you, which we have developed. And let me just skip this slide. This is again the to show that we successfully created chromatin templates from Chlamydomonas, and how I'm going to skip this slide as well, but essentially how we use this template. Okay, the key lies in the digestion stoichiometry. Remember, I told you there are endonucleases that digest the chromatin, So obviously, if there are enzymes that are going to digest the chromatin, if you're looking at an open structure of chromatin, those enzymes will be able to more easily access that chromatin and digest the chromatin much more than compared to a region that is closed and therefore inaccessible to the enzyme for digestion, right? So we decided to exploit this. So this is the hypothesis, and we decided to exploit this and imagine that if we look at a late digest, which is most of the bulk chromatin, and compare them with an early digest, will the early digest be able to differentiate between a more open euchromatic region and a more heterochromatic region? Okay, so how to think about this? Let's look at that. So I'm going to skip this slide, but the idea, what I'm presenting in the slide is that this assay was a versatile assay which worked not only in Chlamydomonas, but also in other yeasts as well as other algae, and was able to generate chromatin templates even from differentiated cells of Chlamydomonas, which is gametes and zygotes. Okay, so Chlamydomonas has a complete life cycle which shuttles between haploid and diploid phenotypes. Okay. All right, so what we learned, I'm going to cut a long story short here because of lack of time. What we learned is that the repeat spacing, okay, nucleosomes are right next to each other, right? So there's a unit of repeat that is repeating. How many base pairs of DNA are wrapped around each nucleosome? If you look at that repeat spacing that we have measured in these assays, you can see that in vegetative cells, it's a certain level, but the moment you gametogenize the cells, those repeat spacing increases. Okay, so what does this mean? And what we have seen from literature is that people have shown, and I can't explain this graph in great detail. This is a very old assay which was developed in 1992 by a very famous person called John Widom, who's a very famous person in epigenetics. And he showed that differentiated cells in sperm, for example, which is a highly differentiated tissue, which is completely compacted chromatin, right? So sperm cells, the length, this repeat length is very high. Whereas in stem cells, this repeat length is relatively lower, okay? Or in undifferentiated cells, this repeat length is relatively lower, okay? And so what we are
Dr. Sen? Hello, Dr. Sen, can you hear me?
Jennifer? Yes, ma'am.
I think Sir's internet is connecting and reconnecting.
Is it mine or is it someone else's?
Yes, sir, you're back.
Okay, all right. Can you hear me? Can you see my screen?
Yes, sir.
Yes, sir.
Okay. So what I was saying is that there is a difference in the repeat length number. And what we know from data today is that this increase in repeat length is a reflection of the shutting down of chromatin. So we used to think that sperm cells have compacted the chromatin because it's a small head and you need to compact the chromatin to fit it into the sperm head, right? But here we have Chlamydomonas, which is a unicellular organism, and the vegetable cell and the gamete cells are not that different in size. They are pretty same in size. And yet the chromatin is being compacted, right? So it is not just because the sperm chromatin needs to be compacted, but this is a very ancient evolutionary mechanism that we have shown that even in gametes which are equally sized, right? In Chlamydomonas, both the gametes are equally sized. The gametes undergo compaction of chromatin. So there must be some method or some reason for this compaction of chromatin, which is later was used for compaction in sperm cells as well in humans. Okay, so this is something that is we are kind of querying in the lab as well, and I'm not going to go into that, but that's another line of fundamental study as to why chromatin needs to be compacted when gametogenicized. Okay, what we did was with this assay was that we separated the mononucleosomes. So when you saw the ladder, right, what was the ladder? The ladder essentially was mononucleosomes. The next band was a dinucleosome. The next band was a trinucleosome, then a tetranucleosome, and so on. Okay, so we separated these bands and we isolated these bands. And what we imagine, if there is an open gene, if it's an open chromatin, the endonuclease will cleave it more often. And therefore, there'll be more number of mononucleosomes coming from an open region. And from a closed region, you will have relatively fewer number of mononucleosomes, more number of higher order nucleosomes. Okay, so this order of nucleosomes is what we started query, and this is a way to look at higher order chromatin structure. Okay, so when we did this, and I'm not going into the details of it again, I'm going to ask you to focus on this particular blue box. Okay, so this is the order of mononucleosomes going from mononucleosome to pentanucleosome, and this is a gene that is being expressed across the differentiation paradigm. Okay, so this gene gets expressed in zygotes. If you notice in the vegetative cells, this gene is completely repressed. It's only enriched in the pentanucleosome. But the moment it goes through gametogenesis and then zygotes formation, it undergoes different transition states of chromatin. And in the zygotes where the gene becomes expressed, the gene becomes completely into mononucleosomes. Okay, so this is the promoter of the gene I'm talking about. This is one single nucleosome we are looking at. So this nucleosome goes all the way from a compacted state all the way to this, and you can actually track. So there's another example here where you can actually track intermediate states of chromatin changes. Okay, like here you can see it's going from the tetranucleosome to the dinucleosome, trinucleosome, and then eventually to the mononucleosome, right? Here it is the mononucleosomes. So we have developed this assay which can now start looking at the three-dimensional structure of chromatin, how chromatin transitions. Okay, how are we using it? And essentially we are looking at many phenotypes in Chlamydomonas, and we are trying to ask how does the environment change the epigenetics of Chlamydomonas? Okay, so where are we looking for this? And these are my last few slides. Please bear with me. I know I've been speaking for a very long time, and I'm going to kind of end on this note quickly. This is the conservation that I'm talking about. And here is a protein that we are comparing between humans and Chlamydomonas, the same polycomb protein, which is EZH2, okay, an enhancer of zest homolog 2. So this particular EZH2, this is an overlap between the polycomb predicted structure of Chlamydomonas against the human crystal structure. And what we see is that there's a very clean, very nice overlap of all the domains, okay? So this means that the chlamyd protein might be actually functionally very similar to the human protein. Okay, so this is something that we are querying, and we don't know how much of this is. This is of course in silico, so we are kind of querying this in vivo. This is an assay that we have created, and what we have learned from this. This is an assay to detect epigenetic changes. Okay, so this is a phenotypic assay which can tell you if there are epigenetically the gene is getting silenced or not. And what we see is that, I'm not going into the details of the assay because that gets too long, but what we have seen from this kind of an assay is that this gene that we're testing for is getting silenced clearly by high zinc and high copper specifically, not by other forms of transition metals that we have tested. High zinc and high copper are very good at silencing genes. Okay, so then why so? So this is something that I didn't tell you about, that this particular protein has 6 zinc binding sites. Okay, so that is interesting that in our assay, the same zinc came up as one of the metal ions that was increasing silencing. Okay, and this is by phenotype. We have not checked these molecularly. We are yet to do that. And another thing was copper. So why copper, right? So we noticed that when cells were exposed to copper, and this is the cells that are exposed to copper, you will see that when cells are exposed to copper, this is an assay that detects ROS, okay, that is able to detect intracellular reactive oxygen species. And we see that whenever there's reactive oxygen species within the cell, those cells will show high green fluorescence. Okay, so the rest of the cells, as you see, there are many cells in this field, but most of the cells are dark, do not have ROS under normal conditions. But and here there are many cells, but here you can see most of the cells that are there in the field have ROS. Okay, so that they are, whenever there's high copper, there's a high incidence of ROS. And not, again, I'm going to skip most of the experiments that we have done, but this is correlating what we have seen here, that both copper and zinc lead to enhancement of gene silencing, okay, for this phenotypic assay that we have generated. So essentially, in Chlamydomonas as well, we have shown that zinc and copper together can lead to gene silencing. And what we have shown is that by using various inhibitors, for example, sodium butyrate, which works in mammalian systems, this is a specific inhibitor against the mammalian protein, designed against the mammalian protein, which is inhibiting called polycomb. And this is another inhibitor which is inhibiting DNA methylation. All these 3 inhibitors work in chlamydomonas. Okay, so this tells you again about conservation of functionality of these mechanisms in chlamydomonas, right? So this is where we are kind of at the stage. We know that ROS-induced gene silencing is a conserved mechanism or conserved pathway even in the unicellular organism Chlamydomonas, which is now manifesting in higher organisms as cancer. Okay, and we are now using this assay as a way to screen for new epigenetic drugs. Okay, and I'll quickly show you how we are going about it. This is again just one last slide. This is my last slide. Bear with me. So this is a known deacetylation inhibitor. Okay, this is a gradient plate assay. I'm not going into the details, but look at the phenotype. Okay, you see that the cells are not growing on this side and are growing on this side, where the concentration is low. Okay, where the concentration is high. Okay, so when the concentration is low, your cells are being silenced. The same gene is being silenced. Okay, this is the control. So the gene is being silenced here, and these are unknown inhibitors, which we had taken cinnamic acid from as a plant-derived compound. Curcumin is again a plant-derived compound. Quercetin is also a plant-derived compound. And we assayed different plant-derived compounds, and I've just shown you some representation here. This particular compound shows a similar phenotype for the silencing, right? So the question was, is this also a deacetylation inhibitor? Because this is a known deacetylation inhibitor in mammalian systems. So our hypothesis is that by generating libraries of Chlamydomonas, these are different clones of Chlamydomonas. This is a library of Chlamydomonas we are trying to generate. By generating these libraries, we can start screening for drugs that will inhibit these epigenetic pathways that are important for cancer. So now we can start discovering new epigenetic drugs in cancer. Okay, so that is kind of the, and there's proof of principle, because we know today there has been a paper published that trans-cinnamic acid is actually inhibiting the histone deacetylases and has anti-tumor effect against colon cancer xenograft in athymic mice. Okay, so we know that cinnamic acid today is actually this was published again right after when we discovered this. This came around the same time when we had discovered this. Okay, so we are now developing this as a tool to look for new epigenetic drugs. So we have this. So I'm just going to thank my funders, which is essentially the Ramalagas Funding Fellowship, all of this work, and Snehal, Pooja, and Patricia, who have done all of this work, and essentially many people who have been involved as summer students and project students in our lab, and currently Sheetal and Priyanshu are working in the lab. Okay, thank you. I can take some questions if there are any.
Thank you, sir, for the enriching and enlightening lecture. We will take up questions now from the participants. Anyone who cannot access the chat box can raise their hand. There are a couple of questions which I will just read it and Sir can answer that. The first question is from Akshay Anand. His question is, if any variegation was observed in the cell or if there was uniform gene expression silencing.
So you have hit the nail on the head, right? So what I didn't use this term in my talk because this is a term that is quite confusing to many people. The difference between variation and variegation. Variation is genetic and variegation is where you maintain the DNA content the same and you variegate the phenotype. So you have the same clone, even in a clonally identical population, you have different phenotypes, right? So that is called variegation. And you are absolutely right that in all our things, what we are actually measuring is variegation. For example, I'll quickly again share the slide just to show you what is the variegation that we are talking about, right? So here, for example, in this particular slide, you can see different clones. These are all different clones in the library, and each of them is getting silenced to a different degree. So these are, each dot represents a different degree of silencing because it's a different dilution. So you can see that some clones are getting silenced less and some clones are completely getting silenced, right? This clone is completely silenced. This clone is getting silenced only in the highest concentration. This one is able to grow all the way to the lowest dilution. So it's not getting silenced at all. So this is the variegation that we see in the population. And the variegation is directly correlated to the epigenetic mechanisms of inheritance. I did not go into those details in today's talk, but that's true. Yes, Akshay, you're right.
There's a second question from Larissa Gomes. In fact, she has two questions. The first is it safe to say plasticity in a cell's function is inherited from generation because of methylation at the specific areas in the gene? Her follow-up question is, does this also mean that cell trauma is transferable through germ cells and therefore to the progeny?
So yes, the simple answer is yes, it is true. The complicated answer is we don't know how much it is for humans. Okay, so the slide that I showed you very early on, one of these, let me quickly go to the slide and demonstrate what I'm talking about. This slide is where I told you about the malnutrition, and I mentioned something called the Dutch hunger. Right now, what is known is that the Dutch hunger is and this was replicated again. These were unfortunate incidents in human history, but we have learned from our mistakes, right? So the Dutch hunger was essentially when the Nazi regime had completely cut off the Dutch people from any kind of nutrition or nutritional supplies that used to come from the south. So they were completely cut off. And for 6 months, they went into starvation because all they could survive was on potatoes, okay? Stored potatoes for 6 months. So you can imagine that they were really, really starved, and there were many people who were already pregnant during this time. So this is where the study, in hindsight now, when we look at these people who were born of the Dutch hunger, people who were in the second trimester, third trimester, or first trimester, we realize that these babies, the second and the third trimester babies, actually are now obese today. And their children who are now coming up are also having some levels of obesity. So we do know that in humans, this can at least, and this is true for controlled mice experiments, where we know that this phenotype can be transmitted transgenerationally to the F2 generation, up to the F2 generation. We don't know in humans how long it can be transferred, okay? This I'm talking about intergenerational transfer. When you think about cells now, in cellular divisions, now cellular divisions are not going through meiotic divisions. Remember that when I'm talking about this transgenerational, there's a meiotic division involved in between. But when you're looking at just the cellular divisions, in cell line studies, the cellular divisions can almost go on, particularly for DNA methylation, can go on almost indefinitely because the CPG has a direct opposite, which is also a CPG. So any C that is methylated is also methylated on the opposite side. So when the DNA replicates, it replicates in a semi-conservative manner, giving both the strands a hemimethylated strand, which is recognized by the DNA methyltransferase and remethylated back to a CP. So it lies in the mechanism of this repression that why it is also so faithfully replicated. So it can actually go on for many, many generations unless altered actively. Okay, so there are regions in the chromosome that are called constitutively silenced, and there are regions in the chromosome that are facultative heterochromatin. The facultative heterochromatin is more plastic, can transition between multiple phases, whereas constitutive heterochromatin continues to remain silenced unless it is sort of goes through a very drastic change. So that is the long and short answer to your question.
Thank you, sir. We'll take up another 3 questions because of time constraint. The next question is from Ms. Shalmali. Her question is, what keeps CPGs at the promoter region from getting methylated in normal cell? Some people are predisposed to cancer. Are there CPGs at tumor suppressor promoter more susceptible to methylation?
So according to our studies, sorry, there was something else follow up to that?
No, sir. No, sir.
Okay. So yes, according to our studies that we have shown that there is some predisposition in genes that are bivalently marked. Remember, I talked about bivalent chromatin in stem cells, and then I connected that to somewhere that is anti-DNA methylation in nature, right? It was opposing to DNA methylation. So the hypothesis, this is again hypothesis, that we think that bivalent, I mean, there is data to support our hypothesis. There is statistical data that suggests that those genes that are hypermethylated in cancers have more bivalency in stem cells. Okay, so there is something that are known as CAN gene sets, and these are defined by several epigenetic studies. We know about 650 genes that are considered to be CAN genes, okay, cancer-specific genes that undergo cancer-specific DNA methylation. And of these 600, about 300 of them have very high levels of bivalent chromatin. So yes, there does seem to be some molecular correlation there. What is the molecular pathway that is actually important for this sort of understanding is not clear to us. There is now. It is true. Yeah.
So your voice is breaking.
Oh, my internet. So is my voice better now? I'll just have to wait it out until the internet becomes stronger and
it's
so
fine.
my voice comes
So
back.
you can go on. It's just on and off. It's okay.
What I'll do is I'll shut my video. So maybe if I shut my video, it will help the bandwidth. Okay. So what I was suggesting is that that bivalency might be one of those factors that can be molecularly looked at for a correlation as to what predisposes us to cancer DNA methylation. Okay, how is bivalency removed so that cancer DNA methylation can set in at those tumor suppressor genes? So it is true that there are some tumor suppressors that are more often hypermethylated in cancers than other tumor suppressors. It is a study that these are studies that are ongoing, and still we are trying to understand the molecular mechanisms. So there is no clear answer to how it happens.
We have Dr. Wajid. He has raised his hand. Sir, can you unmute yourself and ask your question?
Hello, good evening. Thank you, Dr. Sen. It's an amazing talk. I have been really, we are glad to got to hear you. The question that I have is again, it's to do with the nutritional status. Depends on the nutrition. You said there are going to be issues that we might, face in the future, obesity and various other things. What I just want to ask is, now this nutritional, there is a lot of variation that we see within a country or say across the globe in terms of the intake of the nutrients. So is there any sort of a study that has been done where this sort of food would be kind of an ideal, or people who consume kind of bland food, you see people not suffering from issues of all this obesity and all in future, or people who are with a lot of oily, spicy, that sort of food habits, you see people suffering from diseases or cancer and other things. So hope I made myself clear. So
yes, absolutely. Yes. So if you can see my slide, can you? Yeah.
Yes.
So this is a slide that I didn't include in today's presentation, but since you asked about it, I put this on. This is what really ties cancer with nutrients that we eat. Okay, things that we eat, and what you notice are all these things that are there. Folic acid, for example, is given to pregnant mothers, right? Because folate is a common additive for pregnancy. Is a part of many nutritional diets, and then there's other amino acids, and of course there's glucose. The thing to appreciate in this particular slide is that all of these are boiling down finally through the TCA cycle to either acetyl coenzyme A or through the methionine pathway to what is SAM. Okay, SAM is the universal, the sole universal methionine donor in cells. Okay, so SAM is what is also used by DNA methyltransferases to DNA methylate. So question is, so this understanding is, we do know that there is a direct link between these pathways because the balance of these nutrients in the cells will directly affect both methylase enzymes as well as acetyl coenzyme A, which is part of the histone acetyl transferase enzymes, right? So yes, there is a direct link, but what type of diet will give us a good epigenetic outcome is something that is still being talked about. There is one diet that I would like to bring you to bring notice to your audience, is that there is something, a type of diet which is called calorie restricted diet. And this has been studied for about 20-25 years now. Particularly, and this has molecular similarities even in single-celled organisms like yeast. Okay, even when yeast get calorie restricted, they do the same thing. What they do is that people who have calorie restricted diets, what is calorie restriction? Let me just explain that first. Suppose so each of us require a different amount of calorie intake, if you will. Say I might need 2,500 calories per day. Okay, you might need, based on your BMI, your metabolism and everything, your age, everything correlated, you might need, say, 3,000 calories per day. Okay, but what is important in a calorie restricted diet is that you should ideally have more, you should fulfill on a regular basis, have at the max about 70% of your calorie requirements. Okay, 2/3 of your calorie requirements, 77%, whatever, 67%. So you must fulfill only 2/3 of your calorie requirement for the most part. Once in a while, if you do what quote unquote cheat days, it's fine. But people who do these calorie restricted regimes have very low incidences of many non-communicable disorders and lead longer lives. Okay, they have on an average longer lives than those who don't have calorie restricted diets. And in fact, this is again correlated with those who have red meat diets, or all these diets that are heavy fat diet or heavy meat diet. These lead to a lot of oxidative stress because the more of this you're going to eat, the more of it is going to need to be oxidized by your system, because all of we are converting all our fixed carbon to carbon dioxide, which we are exhaling out. And again, that carbon dioxide being fixed by plants, and we are again assimilating them. So all of life is a redox reaction. So the moment the more you consume more calories you consume, the more redox stress your cells will have. So there are these pieces of the puzzle that we know about. How they put together at the molecular level is something that we still don't understand clearly. So our studies suggest that yes, ROS has a direct role in epigenetics because we have shown how epigenetic proteins come together and create these differences in epigenetic outputs that eventually might lead to non-communicable diseases.
Thank you very much, Dr. Sen. Yeah.
The next question is from one of the student, Noinika. Her question is, what are your opinion on the highly debatable topic suggested by various doctors like Dr. Joe Dispenza of self-spiritual healing for several diseases like cancer, etc., through various methods, and its epigenetic proof within the person's lifetime and transgenerationally, and the idea that if environmental negative stimulus, physical and mental insult can create epigenetic changes, then cannot positive stimulus have similar epigenetic effect?
So we understand very little of everything in this question that has been asked. In fact, I would go one step back and tell you that we don't even understand whether many of these diseases that were mentioned, or many of these ailments that are mentioned, have epigenetic basis or not. Okay, so there's most of this question that has been pushed forth is actually conjecture, and of course, everybody wants a solution today, and therefore so this is an argument that is common in science, which is called God of the gaps. Essentially, why the sun rose in the east and set in the west. So, many cultures came up with pagan gods, and that, there's the sun god, a sun chariot that is going to go from one end to the other and drag the sun, and every day this cycle repeats, right? There are many pagans have these sun god rituals, from Egyptian culture to Mayan culture to Indian culture. Many cultures have had that. But today we know the reasons why the sun rises in the east and sets in the west. So do we forget those gods and therefore stop being, quote unquote, dismiss them? That's a faith-based argument. I cannot impose anything on the faith-based argument, but we can for say that there is no chariot pulling God that is going to pull the sun from one end to the other, right? So this is the argument of what is known as God of the gaps. What is God of the gaps is that for every reason that we don't know the answer to today, we tend to fill those gaps with faith-based arguments, okay? And this healing, natural healing or holistic healing or quote unquote, I don't know, pranic healing, or there are multiple things that I've heard in many WhatsApp messages. I see a lot of misinformation going around, and all of them say, oh, they will use very fancy terms. They'll use epigenetic terms and they'll use all these fancy terms and make it sound very scientific. But the difference is in differentiating. Scientific difference for a scientist is to be very conscious when you're differentiating coincidence versus causality, okay? Coincidence can be many things. Every person who drinks water on this planet will die. Therefore, does water cause death? Obviously not. There's a 100% correlation that everybody who drinks water dies eventually, right? So coincidence is not a good way of looking at scientific causality. Causality can be only derived molecularly, and that is what differentiates modern methods of science from the classical methods of science, where we go into mechanism, how things happen. Only if I know the mechanism of the disease, then I will be able to tell you that, okay, maybe doing this type of diet or maybe doing this type of stress busting mechanism might affect it, because the stress busting mechanism, this type of stress busting mechanism that you're doing is affecting this particular epigenetic mark, which is affecting this particular gene. Right now, this is all faff. There is no way to tell whether this is happening or not, right? So this is just coincidence. Causality cannot be drawn from coincidence.
Thank you, sir. Can I now request Dr. Beatrice to unmute herself and ask the question?
Thank you, Dr. Subhojit. A very nice session. I just wanted your comment on how would human microbiome have its effect on epigenetics in the cure for cancer? Hello?
Yeah, I heard that. I'm just thinking because we don't even know whether microbiomes can cure cancer. So I don't know if I don't know how to answer that question, because currently there is no link between changing microbiomes and cure to cancer. Yes, we do know that gut health is very important for many diseases. Okay, and that has, for example, what I showed you in my data is that there was this ETBF treated mice, okay, which is essentially an enterobacter bacteria. It's a particular type of bacteria that causes infection. And Helicobacter pylori, for example, is another bacteria that is known to be highly correlated with cancer. And both these enterobacters and Helicobacter pylori are highly correlated because they cause a lot of oxidative stress in the colon. And oxidative stress in these, so essentially microbiome, if it is going to cause oxidative stress, it will lead to epigenetic changes, and those epigenetic changes might lead to cancer. So all of this, there is a lot of might in this connection, right? There is no direct causality, but yes, there is. What we have shown in our experiments is that there can be that possibility. That needs to be now assayed for.
Thank you so much.
Copy. Can I ask one question to sir?
Sure.
Okay, this is actually with respect to the experiment that you showed, in which you have used the entire Chlamydomonas cells and you have treated them with nucleases and you got a pattern of the thing. So my question is, if you had done the same thing after extracting the DNA, would you expect the same pattern or?
No, because when you purify DNA, you're removing the proteins, you're removing the geography of nucleosomes. So the nucleases are these nucleases are dependent on nucleosome. That part is less accessible to the endonuclease. The endonuclease will go and cleave more often the linker DNA between the two nucleosomes. And that is what leads to the laddering pattern or the repetitive pattern of the DNA. In fact, the lowest band will be a 150 base pair band. The next band will be an exact double of that band, which is about a 300 and something base pair band. Then the next band will be a 450 base pair band. The next band will be a 600 base pair band. So it's like a repeat of 150 nucleotides. And that tells us that the repeat length then would be 150 nucleotides, right? So this is how we measure linker length or repeat length. And that is one of the figures that I showed you that tells us about the compactness of the chromatin. If we can measure this bulk, the repeat length of bulk chromatin in vivo, we can actually measure the compactness of chromatin, both locally as well as globally.
Thank you, sir. Thank you.
Poppy ma'am, you're muted.
I'm so sorry. Thank you, sir. We will take one last question from one of our students. Do you think this is valid? If we make mesenchymal cells to turn back to the epithelial cell, we can stop cancer spreading?
Okay, I'm not clear about the question because...
if we make mesenchymal...
So you're saying... Yeah, because you're saying that basically cancer cells have now more of a mesenchymal phenotype. So if you can reverse this, can be converted to back
yes.
to
yes.
So yes and no. The problem is that most of the cancer cells have become genetically condemned to cancer phenotype, right? They have become genetically sort of ordained in a way such that they will follow the cancer pathway because tumor suppressor genes have been mutated to become inactive, and cancer oncogenes have accumulated oncogene mutations which have made them active, right? So even if you convert them to a mesenchymal phenotype, what will probably differ from mesenchymal to epithelial phenotype, what will probably differ is that you might be able to slow down metastasis. You might be able to slow down the benign growth of cancer. But you cannot cure, quote unquote, cure the cancer because cancer is still now completely genetic. Once cancer has happened, okay, once the tissue has become cancerous, then turning it back is going to be very, very difficult. What you can do though, because, and also remember, cancers are very heterogeneous. The types of mutations that you see are one layer of heterogeneity. What I showed you today in that aging paradigm is that each cancer has different epigenetic heterogeneity. So even a single benign tumor has a lot of heterogeneity, even epigenetically, which means that even within the same tumor mass, some cells will respond to a drug differently and than the other cells. And that is what really gives rise to a lot of resistance as well, right? So this is kind of an evolutionary process if you think about, but evolution not in the scale of time the way I have talked about it, but evolution within the body of how tumor cells are evolving, right? So we do know that that studying these processes is going to tell us, this EMT and MET processes is going to tell us something about how to treat metastatic cancers or how to deal with aggressive cancers. Can we stop aggressive cancers? But I don't think it would be in my opinion, of course, it's not going to be a cure because you're already genetically committed to a type of cancer. That's the long and short of it, yes.
We will wind up the Q&A session now due to time constraint. There are a couple of more questions. We can mail it to Dr. Shubhajit. Sir, I thank you again for answering the question with so much of patience and clarity. Thank you so much again. With this, I hand over the platform to Madiha. Over to you, Madiha.
Thank you, ma'am. I now call upon Shivani to deliver the word of thanks.
A very good evening to one and all. It gives me immense pleasure to deliver the vote of thanks for this inaugural event. First and foremost, we must thank God who with his benevolent gaze has given us the strength and the drive to hold the first meeting for this academic year. On behalf of the Microbiological Society at St. Joseph College, I would like to thank the speaker, Dr. Subhojit Sen, for enlightening us on his life's work in such a clear and interesting way. We could clearly see the relevance of such a topic, especially in this modern paradigm, and hopefully this motivates people to pursue a career in such a field. I would also like to thank the Rector, Father Shubhajit De Silva, Principal, Father Dr. Victor Lobo S.J., Registrar, Dr. Melvin Colasso, and our Vice Principal, Dr. Rabai, for providing such an amazing platform to showcase our interest and topics to one another. This event wouldn't have run like a well-oiled machine without the help of our Head of Department, Dr. Beatrice Siqueira, and our postgraduate coordinator, Dr. Poppy Datta. We thank them for their continuous support and for proving the successful mediation between the students and the college administration. I would also like to extend my thanks to all faculty members of microbiology, namely Dr. Vanita NM, Dr. Syed Wajid, Dr. Jyoti William, Ms. Riya Shah, and Ms. Shalmali Kamath, who have taken up the arduous task of organizing and planning this event under the current challenging circumstances. Thank you, Madiha, for being such an amazing MC. There wouldn't have been a better choice, and I certainly hope that you would do this more often. Not only her, but all members of the Microbiological Society must be lauded for working tirelessly to make this event possible. I would also like to thank Shankar for YouTube live streaming. Lastly, this event would be meaningless without our audience who have come for this event. We hope that we have achieved your expectations and have sustained your interest. We look forward to the continued interest and attendance at future events of our society. Thank you.
Thank you, and all the best to the association.
Yes, thank you. I think with this, we've come to the end of our session. So the live stream will end shortly. So I request the participants to log off now. But I do request the office bearers, the faculty, and sir, please turn on your cameras so that we could take a picture for this event. We usually would have done this, but considering the situation, let's try to do the best that we can. Even Dr. Melvin, if you're here, if you could please turn on your camera.
I think Dr. Melvin is not here. He has another meeting, so he has left a few minutes back.
Okay.
Sam, you are going to take the picture?
Okay, ma'am, I'll do it. Just give me a second. Yeah, that should, okay, wait 1 second. Beatrice ma'am is coming. Hold on, ma'am. Okay. Everyone say cheese. Yeah, ma'am, yeah.
Thank you, Sam. Thank you.
Thank you, sir, and thank you all. We hope we meet again.
Yeah, I hope.
Yeah, thank you.
Yes, sir. So please let us know when you come to Bangalore. We would like to invite you to our college.
Sure, I would love to come and see this. Yeah.
Thank you,
Please.
sir.
Thank you.
Thank you all. Thank you, dear students.
Thank you.